Hello, my dear students. Hope you all are fine at home. We are going to continue the lesson evolution. So, in the last video, we have discussed about the Hardy-Weinberg principle, or it can also be called as Hardy-Weinberg principle. So, this equilibrium or principle states, what does this principle or this equilibrium states? It states that in a population, okay, so we can find out the frequency of alleles of a frequency of occurrence of alleles, okay. So by this principle, using this principle, we can find out the frequency of occurrence of alleles, okay, in a gene, okay. So that is what the purpose of this principle. So the definition is that allelic frequency in a population are stable and it remains constant from one generation to the next generation. So mainly here the allelic frequency remains constant from when to when, from one generation to next generation in a sexually reproducing organisms. Okay, there are some conditions are there which based on that condition only we can assume this principle or this principle or this equilibrium can be applied if those three conditions are existing. So we'll go to that, we'll discuss about that conditions for this Hardy-Weinberg principle in the next afterwards. So next the equation states that p square plus 2pq plus q square is equal to 1. Remember, this is a part of population genetics. In the last video, I told you about the population genetics means that study of the frequency of genes in a population. So, population genetics is a part of biology which relates with the study of frequency of genes in a population. So, we can find out the particular characteristics, how much it is continuing, or which character is more dominant and which character is more seen in a particular environment. Those features can be calculated or can be identified by using this principle. So generally we know that this frequency remain fixed. Okay, the frequency, the occurrence of these alleles remains fixed throughout the generation. So here, this individual, we say that in a population, the gene pool remains constant. So what is the gene pool means? It's a sum total of genes and their alleles. What is this gene pool means? I told it also in the last class already. Sum total of genes and their alleles in a population is called as gene pool, and that it also remains constant in a given condition. So in a given population. Then we have assumed here, actually this is a application of mathematical probability. You know that it is based on the mathematical probability, and also it is an application of Mendelian genetics to the Darwinian natural selection. So Darwin so far he said only the natural selection variation and all. He did not use the genetics part of for this. So after the Mendelian principles, later this principle was developed. So we can say that it is application of Mendelian genetics to the Darwinian theory of natural selection. So in a nature or in a generally an individual, that diploid individual will have two sets of alleles. A character is considered by the two alternative forms of genes. That also we discussed. So what is that two alternative forms of genes? We have considered the consider the character A and the small a. So we have selected the genes as A and A. Then alleles will be the capital A, capital A. And we know that in a diploid individuals, they may remain either in the homozygous dominant condition or then homozygous recessive condition, or they can also remain in the heterozygous condition. So by adding up all those we can understand that. Okay, so by adding this homozygous alleles, then heterozygous alleles, then as well as then dominant recessive, then we will get that equation as p square plus q square plus 2pq is equal to 1. Okay, so this is an application of mathematical binomial theory you might have learned. Okay, it is an binomial expansion of p plus q whole square. Okay, now what is the significance of this equilibrium? What is the significance? This significance is that it helps for the population genetist to assess the degree of evolutionary change. Okay, what is the amount of evolutionary change occurred in a particular population by comparing the allelic frequency of present generation to the next generation. This is for the significance. In total, in a short way, just it helps for the analyzing the population evolutionary change. Okay, then we also understood about the factors. Okay, now in detail we are going to study about that. Now let me tell about the conditions assumed for this Hardy-Weinberg principle. So the conditions, in which condition this principle can apply, it is the conditions like if the population are large enough. So only in the large sexually reproducing population we can apply this principle. Then second condition is that no mating occurs in a random manner. No mating occurs in random manner. That also should be satisfied. Okay, that means in a population. So by what is population means? Just take down this population definition once. It's a group of population means it is a group of individuals of same species. It's a group of individuals of same species of same area at a particular time. You may be thinking population, who knows? It is a very common term. Everyone knows this. But when you are writing the definition in a biological manner, you must use some of the terms. Okay, it's a group of individuals of same species of same area at a particular time. That is a population means. At a particular time, what is the number of a tiger species? What is a number of a lion species? Or what is the number of particular insect or same species? So then only we can say it is a population of that insect, population of tiger. So that is what the population means. So second condition, we'll come back to this. The conditions assumed for this are the populations are large enough, then no mating occurs in a random manner, and third condition for this is no mutation occurs. No mutation occurs or does not take place. No mutation takes place. Okay, if the mutation takes place, then it should be at same rate in both the directions. No mutations takes place. Okay, if this Hardy-Weinberg principle or equilibrium can be applied if there is no mutation takes place. If mutation takes place, the rate of mutation is same in both the direction. The rate of mutation is same in both the directions. Here, what this direction means, It means that even the number of new population as well as number of the old population, their frequency must remain same. Okay, so mutation, if it should not, if this condition cannot be satisfied, this equilibrium cannot be maintained if there is a mutation. If in case mutation occurs, there should be a balanced mutation where both the mutation occurs in the same rate. Then third condition for this is, yes, it is fourth condition, no migration or no migration should take place. That is, population must remain constant. So that is, no immigration or emigration, and it takes place, that is, in a closed population only we can apply this Hardy-Weinberg principle. In a closed population where there is no migration takes place, there this condition can be satisfied. In a closed population where there is no migration, no immigration as well as no emigration, then only this condition can be applied. So these are the conditions assumed for this equation, that is, populations are large enough. Second point is that no mating occurs at random manner. Third point is that no mutation takes place. If the mutation takes place, the rate of mutation is same in the both directions. Then fourth condition is assumed for this is that it takes place. This condition can be applied in a closed population where there is no migration takes place. Okay. Migration can be of two types. What is the two types of migration? Either immigration or emigration. Some population can be added, some new species can be added to a population, or some of the species can move out of that place. So there will be a change in their population size. So if that migration is there, means then that equilibrium cannot be maintained. Okay. So this is about the Hardy-Weinberg principle. Then also we understood if that number, okay, if this is not equals to 1, what does that indicates? Okay, it indicates that the evolution has occurred. Okay, if there is any disturbance in this equilibrium, it indicates that the evolutionary change. Then also it will be we can also, this equilibrium also helps to learn about the occurrence of evolutionary change. Okay, so significance, one more, already one aspect I told you, it helps the population genetics to understand the evolutionary change. Then also one more thing is that direction of evolution, in which direction the character has been, or in which direction the evolution has occurred, or which characters got selected during that evolutionary period. Then also evolution of a selected trait. Okay, we can also assess which character has got selected, in which direction the evolution has occurred, and occurrence of evolutionary change. So these are the things we can assess by using this Hardy-Weinberg equilibrium. And in higher level of questions and all, you can also expect some of the problems by giving this some values for this p square, q square and all. So for NEET and all, you have to learn that also. So go through that part also, how to calculate the frequency by using the Hardy-Weinberg principle. Then genetic equilibrium serves as a baseline against which we can measure the genetic change. So in overall, we can say that Hardy-Weinberg principle, otherwise we can say it is a genetic equilibrium, serves as a baseline. It serves as a baseline against which we can against which we can measure the genetic change, against which we can measure the genetic change. So overall, this is what the main significance. Okay, main significance means it acts as a baseline by which we can measure the genetic change. This is what the main significance. Okay, and let me move to the next part of the Hardy-Weinberg principle. That is the factors affecting the Hardy-Weinberg principle. Okay, when the question comes for 5 marks, it is expected that you have to include the equation, then derivation, and as well as the factors affecting it, the Hardy-Weinberg principle. Just I am writing factors affecting it. So what are the factors affecting it? It is gene migration or gene flow. We can write it as a gene migration, or you can also name it as gene flow. Then second factor affects is genetic drift. Then third factor is a mutation. Fourth factor is a recombination or genetic recombination. We can mention just as a recombination also. And fifth one is a natural selection. Okay, so these are the 5 factors affecting the Hardy-Weinberg equilibrium. That is gene migration or gene flow, then genetic drift, then mutation, then recombination and natural selection. Let us understand one by one how these factors affects the Hardy-Weinberg principle. So first one is genetic. Yes, it is gene flow or gene migration. Students, one thing you notice here in the word itself there are some terms are there which means the almost the same. Okay, here that itself the meaning of that. So just relate with that. Gene migration and gene flow means migration, moving out of the place, from their place. So likewise, if the population moving, so by chance or for any reason, some of the population will get moved. They will get shifted from one place to another, or due to some natural disaster also they may migrate to another place. So this gene migration can also, so otherwise that affects the frequency of that alleles in that particular population. So this factor, if that there is a migration occurs in a particular population, then the equilibrium cannot be maintained. Okay, so this will affect the equilibrium of the individual that frequencies. So first one is gene migration, or also you can take it as gene flow. So the definition is that when migration occurs, okay, when gene migration occurs, gene change, gene frequencies change. Where it will change? In the original as well as in the new population. So when there is a migration occurs, okay, gene frequency change in the original population as well as in the new population. So consider this is a population A and this is a group of individuals. That means where some species are inhabiting. So suppose some species from here they are moving out of this place to the other place. Okay, so here consider some species are there which migrates to this place. And so what happens here? Here already some other species also will be existing. So here the new population will get added, right? And there will be a loss of some genes in the original population. So new genes will be added in the new population. New genes or alleles are added to the new population. And some genes are lost from the original population. So from the original population there will be loss of some genes, whereas in the new population there will be addition of some new genes. This is what the gene migration or gene flow means. Gene flow. So what is the difference between this gene migration and gene flow? So that gene migration occurs in many times. If that gene migration occurs many times, then it is called as gene flow. If the gene migration occurs many times, then it is called as gene flow. There would be gene flow. When will be the gene flow? When migration occurs, when gene migration occurs many times. If the gene migration occurs many times, then there will be a gene flow will occur. So that is what the gene flow means. Gene migration means a particular change. Okay, that means there is a change in the gene frequency if the particular population moves out of their original population. So what happens if this gene flow occurs? Okay, if this gene migration occurs many times, then it results in the gene flow. Then second aspect is that, what is the second factor which affects the Hardy-Weinberg principle? The second factor is genetic drift. Second factor is genetic drift. So what does the genetic drift means? Change in, okay, exact definition is sometimes the change in allelic frequency is so drastic. Change in allelic frequency is so drastic that new population. So sometimes the change in allelic frequency is so drastic that new sample of population form a variant with a different species. So this is what the genetic drift. So I'll make you clear with an explanation. See, so if the sampling is made in a random manner, okay, so what happens is sometimes it is changes occurring. So otherwise we can also say that, in short we can say that the changes in gene frequency occurs by chance. Okay, the gene frequency, there is a change in the genetic frequency occurs by chance. Here for example, in a suppose one time when we considering a population, we could see there is a many type of individuals were there. Just consider, I'll use the yellow color and the okay. Several generations were there of random sampling. So next, when again it was assessed, here the several generations were there. And when it is second time it was assessed, there will be only a few individuals were seen. That means we can take it as almost a 5-6 individuals were there. So in the second time when it was analyzed, there was how many generations were there? Only 6 of the original species or original alleles. Alleles have left. So only the 6 were left. Now again it was analyzed. They could see that there was a completely a new individuals, and only the 2 of the previous alleles were left. So here we can see only 2 of the original alleles were left. Only 2 of the original alleles. So beginning it was many, it was there. Then it came down to 6. Then again it has come down to the 2. So only 2 of the original alleles are left. So this means that there could be any change in the genetic frequency occurs due to the chance mutation. Okay, the reason, what is the reason for that? Completely a new type of alleles to occur in the third generation. That is reason is that when they analyzed for the third time that population after by collecting doing a random sampling, the reason was there was a chance mutation. So due to changes in the allelic frequency in a population, some form a different species. So why there is only 2 were left and all others were new species? Because here some changes has occurred in the genetic frequencies, okay, that formed a different species. Okay, here the effect is called this effect and the original drifted population is called as this population. That is the original drifted population is called as founder. The original drifted population is called as founder, and the effect is called as, and this effect is called as founder effect. And this effect is called as, question used to come, what is founder effect? So you should use this explanation. Okay, need not to use a diagram and all, just use the sentence and write. So what is the definition for this? Sometimes change in allelic frequency is so drastic that new population form okay, new population sample of okay, I just repeated the 2 terms again. Sometimes the change in allelic frequency is so drastic that new population sample form a variant with a different species. So you can see from the original, comparing the original species to here, there was only the 2 species were left in the new population. When it is sampling was done for a many times, beginning there was many were there, again next there was a less it was there. When it comes to the third sample, they could see there was only the 2 of the original species were left behind, and reason was that there was a